Hey there, coming to you live from my basement on a Sunday. Just doing a quick review of all the organelles for you guys in case you need to hear me talk about it one more time. On this slide, just a quick reminder that when we say cytoplasm, we're talking about everything that's inside the cell. Generally, we'll say that the nucleus sits inside in the middle of the cytoplasm, but generally that's where all the organelles are going to be found. Don't be confused by the word cytosol and cytoplasm. Cytosol really refers to the jelly, the liquid jelly-like substance that's between all of the organelles, and that's where we're going to find all dissolved nutrients and that sort of thing that the cell is going to need to either work with or expel. So, and of course all the organelles that we're going to go through right now, so you can have a good handle on that before the quiz. So first particular organelle we need to talk about is going to be the nucleus. Most important thing about the nucleus is that we're going to find our DNA there. There are two other organelles that we do find DNA, and those are going to be the mitochondria in animal cells and the chloroplast in plant cells. But for this is going to be any of the DNA that we use to code to make proteins, which will be made in the cytoplasm. We'll get to that in a couple minutes. So all the DNA is basically kind of stretched out into what we call the nucleolus. It's a dense region inside the nucleus where we find the DNA. These structures that you see here, these chromosomes, only form when the cell is about to divide. So when we talk about mitosis later in the year, we'll talk more about how this happens. It's called tertiary coiling. We'll go through that whole thing. So in terms of other stuff that's on the page, whenever DNA is uncoiled, we call that chromatin. When it coils up, we call it a chromosome. Kind of a silly thing that they have different names, but they're different structures at that point. The other thing that we know is that on the nuclear envelope or the nuclear membrane, we have all these little pores, and those pores are for when we copy DNA. Remember, the DNA never wants to leave the nucleus, it stays there. We take the information from the DNA and we shoot it out into the cytoplasm where all the building materials are, and that information that's copied in the form of what we call messenger RNA, which we'll talk more about later, can shoot out through one of these pores and then head out to the rough endoplasmic reticulum and the ribosomes. Okay. So again, two major functions of the nucleus. One, controls cells' activities, and basically what that is going to be building proteins. That's the number one thing we'll talk about. We have a whole other unit on that later in the year. The other thing is it obviously is going to contain all of our hereditary information. So when the cell divides, all that information will be copied and passed off to the daughter cells. Beyond that, we talked a bit about the cell membrane. We'll go into that more after Thanksgiving. But the cell membrane is this thing called the fluid mosaic model. And so this diagram, which is not the best diagram I've seen, will have these proteins which we call integral proteins. Integral proteins happen to be sitting within the membrane like this. If they're peripheral proteins, they really sit right on top of all these little guys. And remember that each one of these structures that we see, this guy here with the two legs, those are going to be fatty acids. This thing at the top is going to be part of our phospholipid. It's the phosphorus head, okay, the phosphate group that's there. It's a polar compound, so the blue parts that are the heads like to be around water. The yellow parts, which are these hydrophobic tails, don't like to be around water. So the two tails stick together because there's water outside the cell and water inside the cell. So we get this bilayer, okay, the phospholipid bilayer. Some of these proteins we'll learn soon actually are like little channels where things can go through them because they may not want to go through this hydrophobic portion of the membrane. So sometimes we have hydrophilic amino acids that happen to be lining the inside of this protein. Therefore, the things that are polar can get through the membrane rather easily without having to go through this nonpolar section. If you remember, the fluid mosaic model means that all of these things are sliding back and forth and around each other, but we never see these guys kind of flip over backwards and move to this side, or we never see any of these things flip back over to here, because that means that this polar head would have to go through this nonpolar region. So everything more or less is going to move horizontally around this thing. In class I made mention of something called like a psychedelic soccer ball. So you can imagine these patches kind of sliding around each other, and that's exactly what happens here. And there are some other things not in here. We do see there are other proteins and carbohydrates that are present and other lipids that are present on the membrane. Remember, we talked about the fact that cholesterol is in here because cholesterol actually is going to keep the membrane somewhat fluid. Without it, it becomes a little bit too rigid, and so this is actually going to keep it a little bit more fluid. Okay. The cell wall, only found in plant cells and some bacteria cells and fungi, is going to be something that's rather thick. It is something that doesn't really have a lot of give to it. It's there mainly for structural support. In class we had mentioned before that the cell wall actually has a couple different parts depending on what kind of cell it is. We have what's called the primary cell wall, which is the first thing that's produced, and that is the thing that is sort of on the outermost layer of the cell. And And then inside that we see the secondary cell wall, and that actually can be rather thick. And if you look at this whole section right in here is all going to be the secondary cell wall. Whereas way back here, closest to the cell itself, is going to be the cell membrane. Remember, the cell membrane has a totally different job. Cell membrane lets things in and out. Really, these other two things are here for structural support. We'd mentioned in class that this stuff right here, which is going to be our secondary cell wall, has this stuff in it called lignin. And the lignin is something that has a lot of structural support. So when you think of wood, something with woody stems has a lot of cells that have that lignin. Outside of the secondary cell wall, we have another thing called the middle lamella, which is essentially a layer of this stuff called pectin. And pectin is kind of like a glue that's going to keep those cells real close together, kind of glue them together to maintain strength for the plant. And so when you think of, I think in class we had talked about trying to break a board with your hands, like you're a martial arts expert, and that if you want to do that, you really need to be able to break the pectin that's between there. Okay. So on to mitochondria. Mitochondria, we talked about, kind of looks like, I don't know how people say this, but it looks like a hot dog with a bunch of like a maze inside of it. I think the thing that I made mention of in class that's important is that all of these membranes that are here that are folded increase the surface area of the inside of the mitochondria. And we haven't learned about it yet, but there's a reaction that takes place on those membranes that actually creates ATP, adenosine triphosphate. So those membranes, because there's a lot of them, means we can make a lot of ATP. So the more membranes we have inside the mitochondria, the more ATP we can make, which is good because ATP basically powers everything in your body. Okay? So inside that second membrane is an area we call the matrix. The outer part of it, in between the membranes, we just simply call the intermembranous space. Some of these other terms that we talk about, these cristae and such, we'll talk about those when we get into cell respiration a little bit later in the year. Last thing is, you might expect mitochondria, for example, because they produce ATP, cells that are in areas of the body that need more energy, say muscle cells, you would find far more of these things in muscle cells than you would in, say, a skin cell or something like that. Okay, so we talked about the fact that they take glucose and make ATP. Do not tell me it's the powerhouse of the cell. That doesn't mean anything to me. We did also talk about in class the idea that mitochondria have their own DNA. It's thought that they are one of the first things that moved into a prokaryotic cell to create a eukaryotic cell. And in doing so, it has two membranes around it, which is where we think that came from. It has its own DNA, so that makes sense it could have been its own cell. But basically their job is to produce ATP for the cell and for the body, so we can do stuff with that. All right, plastids, something we find generally just in plant cells, some algae. Basically what we're going to see is it's got two membranes, so it's similar to the mitochondria in that case. The two that we had talked about in class are the chloroplast and the chromoplast. The chloroplast happens to be a pigment, or it's an area where we have a pigment called chlorophyll. Chlorophyll happens to be a pigment that absorbs mostly everything except for green light, which means the green light gets reflected. Chromoplasts do virtually the same thing, but they absorb different colors of light and reflect different colors of light. So chloroplasts absorb most things except for green, which gets reflected. These absorb most things except for red, yellows, and oranges. Those get reflected, and that's why they look that color. We talked about in the fall, chloroplasts turn to shut down or denature first, which means these are active for a short period of time afterwards. That's when we see the leaves change color in the fall, because these things are still functioning, while this one has been basically taken apart. Okay. Function of these guys is to take sunlight, carbon dioxide, and make sugars. Oxygen is a byproduct, which is great for us. That's a picture of a chloroplast. These little guys inside here are called thylakoids, and we'll talk about those as the thylakoid membrane. We'll get more into that when we do our photosynthesis unit, which is coming up after this next test. Vacuoles. What you probably see here, this is a plant cell, and I know it's a plant cell because this vacuole is enormous. Plant cells have what we call a central vacuole. It is most of the cell, and that's because plants obviously can't get up and get a drink when they're thirsty. They can't get nutrients when they need them. They just have to get them and hold on to them. So these vacuoles are quite large inside plant cells. They make up the majority of the space. Animal cells, when they're there at all, tend to be very small if they have vacuoles, because we have circulatory systems. If we get thirsty, we can get something to drink. If we need something to eat, we can go get it. So essentially the vacuole is basically going to be a plasma membrane, which is the phospholipid bilayer, surrounding an area that has all of this stuff inside of it. Okay? Later in the year, we'll talk more about contractile vacuoles, probably at the end of the year when we do a little bit of comparison between different types of animals. The endoplasmic reticulum, of which there are two kinds, basically is a transport system for the cell. So the cell has all this cytosol in it with all these nutrients, and there's a lot of things getting in the way. If you want to think about the hallways during passing time being real crowded and hard to walk through, if you had your own secret tunnel that you could hop into and get to your next class, you could get through without bumping into stuff, without interacting with other people. You could get there a lot quicker. That's essentially what the endoplasmic reticulum do for the cell. It gives it a place for these things to get through from one place to another without interacting with things. In the case of the rough endoplasmic reticulum, that's important because these are the things that have the protein factories on the outside, the ribosomes. As soon as the proteins are produced on the ribosomes, they can travel through the endoplasmic reticulum off to the Golgi bodies, which, if you remember, that's where the proteins are going to get kind of modified. They're going to get folded up, they're going to get labeled for where they need to go, and then they're usually sent out of the cell in most cases. Smooth endoplasmic reticulum do not have the ribosomes on the outside, so they don't have anything to do with protein synthesis necessarily. They do have something to do with building lipids. They can neutralize poisons, which also has something to do with lysosomes in some cells, and largely that is their job. Okay, so you also remember that we talked about the fact that the rough endoplasmic reticulum is connected to the nuclear membrane, which makes sense because inside the nucleus we have DNA that is going to be copied into RNA. The RNA is the information that we need in order to build our proteins. That information squeezes out through the nuclear pores and into the rough endoplasmic reticulum, where it can hit a ribosome and we can make our proteins. And that will fold its way out all the way out to where the Golgi bodies are. So ribosomes are made of something called RNA. We'll talk more about this probably in February. But two big RNA subunits called rRNA are going to make up these two big subunits, and essentially this is the place where proteins are made. We'll talk about this in far more detail later, but at this point, know that ribosomes are going to be the place where proteins are produced. These things are going to be found in both animal and plant cells, as well as prokaryotes. It's not a membrane-bound thing, so when we say membrane-bound organelles are found in eukaryotes, this is not membrane-bound. So they happen to be slightly different in prokaryotes, but they're there. Okay? Golgi bodies, weird name on this guy, and remember it could be Golgi bodies or Golgi apparatus. Occasionally you'll see Golgi complex. It's all basically the same thing. It's a series of these membranes that look like pancakes. They're kind of stuck unevenly on each other. Their job is essentially to take proteins that have been produced, the primary structure, which is the sequence of the amino acids. There may have been some primary coil or some secondary coiling in there to make these alpha and beta structures. As soon as that's done, those things will go off to the Golgi body, where they'll get folded up into their shape. They'll be shipped out of the cell if need be, and that's through these little guys called vesicles, which are essentially little cell membrane bubbles. And that's all these things are made out of. So if a little piece of this kind of bubbles out, shoots out to the membrane. When it gets there, it'll open up the membrane and kind of release that protein to do whatever it needs to do. Lysosomes. So lysosomes are things that are used basically for digesting stuff. And it's important to understand that these would be very, very populated inside of a white blood cell, because white blood cells, their job is basically to eat things that don't belong in your body, and that being bacteria cells for one. So once that bacteria cell is ingested by this white blood cell, we need to make sure that it's broken down immediately, because the bacteria cell probably harbors DNA that, if left unchecked, could get into other cells and cause disease. Or if it's could be something that could be transmitted via a pili to something else. So these things are going to be eaten, and when they do so, that stuff can be recycled, and we can use some of those nutrients for other things. So we have lysosomes to help us take care of that. Microtubules, they are hollow structures that are made of proteins. We talked about protein as being a very important thing for structures when we did that stuff. Microtubules are hollow, so it makes them very strong. They're lighter weight. They basically give the cell a bit of structure so that it just doesn't flatten upon itself. They also play a role in letting organelles be transported around the cell a little bit. If you want to think about it kind of like train tracks, you can set the organelles on these train tracks and they can slide around the cell if need be. They're also an important structural piece for flagella and for cilia, which are the two things that kind of move the cell around in a liquid environment for some things. So the protein that's there, of course, is this thing called tubulin, and that's really the only one you're going to need to know by name. There are some other things we could learn about, but we're going to save that for later, if at all. And I believe the last thing we had to talk about in terms of our organelles are going to be flagellum and cilia. And for flagella, it's a big whip-like tail that kind of moves back and forth. Sometimes cells can have multiple flagella. This particular one only has one on it. We saw with bacteria, they could have tufts of them hanging off the side. Sometimes they're all over the place. It's there basically for movement, okay? So these microtubules kind of spin around in circles, which makes this thing move back and forth, and it whips its tail back and forth. Cilia, on the other hand, there's a lot more of these on the outside of the cells, and you can think about it, oars in water, but remember we talked about in class, if you're trying to row a boat and you can never take your oars out of the water, when you move the oars back and forth, basically what you're going to see is this cell kind of move up and down and up and down, and eventually it'll start moving one way or the other, but the flagella is a far more efficient means of locomotion than these cilia are. But it's important to understand that a lot of these cilia can also be found on cells that are stationary inside your body. Example we gave in class was that in the inside of your trachea, inside your throat, these cilia that are there actually help to kind of sweep out some of the junk that gets inhaled down into your lungs. And so when these things are moving, they effectively take stuff out of your lungs, move it up in your throat, you cough it out, and that's some of the phlegm that you'll cough out. We had mentioned that smokers, for one, whenever you smoke a cigarette, it for some period of time will sort of paralyze all those cilia so they can't move. So any of the chemicals getting down in your lungs tend to build up in there until they wake up and they start to sweep them out. So we talked about smoker's cough with people in the morning waking up and sounding like they're hacking their lung up because a lot of these things have woken up and they're trying to get all those things out that were inhaled into the lung. So in terms of stuff we have to do for this next test, that's about it. Or excuse me, for the next quiz. We'll talk more about some of the other things the next time I happen to do one of these. So that's about it for now. If you have any questions, make sure you see me. Thanks.